Hello and welcome to episode 611 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and I am looking forward to sharing our interview with our guest, Dr. Andrea Califano, originally published on May 1st of 2015. Listeners, Andrea has done groundbreaking work in the field of systems biology, and his work has helped identify, at the single-cell level, the molecular factors that are involved in tumor development, cancer progression, and the emergence of drug resistance. Through his research, a number of master regulator proteins have been identified in cells, and these proteins are now being examined to determine their potential as therapeutic targets to better treat cancer and drug resistance. So listeners, sit back, relax, and I hope you're ready to learn more about Andrea and his work and to get inspired in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Today, I'm excited to introduce you all to Dr. Andrea Califano. So, Andrea, thank you so much for joining us today. How are you?
I'm doing great. Very happy to be on the show.
Wonderful. We are thrilled to have you with us today. And listeners, just a little bit about Andrea. He is the Clyde and Helen Wu Professor of Chemical Systems Biology, Chair of the Department of Systems Biology, Director of the J.P. Sulzberger Columbia Genome Center, and Associate Director of the Herbert Irving Comprehensive Cancer Center, all at Columbia University. He completed his doctoral studies in physics at the University of Florence in Italy and his postdoctoral studies in physics at MIT. Afterward, Andrea worked at the IBM T.J. Watson Research Center, and he subsequently became the program director of the IBM Computational Biology Center. In 2000, Andrea co-founded First Genetic Trust, Incorporated, and founded another company called Therasys in 2008. Later, he joined the faculty where he is today, at Columbia University. And now, Andrea is here with us today, listeners, to tell us about his journey through life and science. So, Andrea, we are excited to get to know you, not only scientifically, but also see a little bit of your life outside of science. So can you start by telling us what you like to do with your time when you're not doing science?
It's a great question. I don't have a lot of time when I'm not doing science, unfortunately. And also having four daughters, age 5, 9, 19, and 21, plus my wife, really creates tremendous pressure to do a lot of other things that are very social and exciting, like going to the botanical garden, the zoo, or maybe going to listen to some music. My passion when I really have free time also to spend with my family are playing tennis and skiing in terms of sports, but also I'm a big movie buff and do an enormous amount of reading.
Fantastic. So what's the best movie you've seen lately, Andrea?
Best movie I've seen lately. They all blur together. It's very difficult to say. Actually, I just saw a movie on TV called A Most Violent Year, which I thought was really extraordinary, well done. It was a story about New York in the 80s and how sort of a businessman who's not completely fully straight, but tries to be as straight as they can, sort of comes of age and fights off the rest of the more corrupt businesses. It's a very interesting movie. I really enjoyed it.
Well, I haven't seen that one. I'll have to add that to my movie list. Fantastic. So great to hear about your life outside of the lab. But next, I want to talk about your science. Can you tell us how you describe what you do to someone who's not familiar with science?
Let me try to make a metaphor for what I do. What is the first thing you do when you're trying to fix a dishwasher that is not working properly and you don't have the opportunity to call the actual technician?
Oh, that was going to be my first answer.
That always works, but let's say there's no reference centers. Your phones are dead. What are you going to do? The first thing you do, you try to actually go either on the internet or in a specialized bookstore and buy a blueprint of how the dishwasher works and what are the parts and how they're working together. So in biology, we have done an amazing job of actually listing all the different parts, all the different genomic, proteomic, metabolomic parts that work in the cells. But by and large, we have absolutely no idea of how they work together, or at least the ideas of how they work together are extremely primitive. And so what we're trying to do is to actually reconstruct from scratch the mechanics of the cells, if you want, the assembly manual of the cell. And unfortunately, like a Miele dishwasher would be different from a GE, all cells are different. And so their regulatory networks and signaling networks, which are basically what defines their logic, are in fact extremely different. And so what we've been doing over the last 10 years has been to reconstruct models of regulation in the cells that describe very precisely not just physiological normal cells, but actually more frequently than not, cancer-related cells, and actually cells involved in a variety of other diseases, including neurobehavioral and neurodegenerative diseases such as ALS and alcohol addiction, etc. And then once we have these models, we can actually ask questions in a very different way. Instead of asking, as most people do, what are the things that change between health and disease in a cell, or between two states of normal physiologic behavior, we can ask what are the proteins that actually regulate what changes in those conditions. So these proteins end up being the generals, if you want, that are responsible for implementing the plans in the cell or responding to certain insults like mutation in cancer or responding to certain environmental challenges.
Very interesting. I like how you start off your story of your research with this analogy, this metaphor about the broken dishwasher, how to fix it, and kind of getting those details about the cells and what's going on in our own bodies, which is very cool. So we'll talk more in detail about your research as we go through the interview. But I want to stop here and have you give us a little piece of motivation or inspiration. I'm always looking for motivation, especially early in the morning here. So can you share your favorite motivational or inspirational quote?
I think that actually because this is a time of great challenge for science in terms of funding, one of my favorite scientists is Sir Rutherford, who actually discovered that the atom is not full, but that most of the atom is actually empty, and there's a little tiny thing in the middle called the nucleus, and then these electrons spinning around. And one of his most famous quotes is, "Gentlemen, we just run out of money. We now have to start thinking." And I thought that was just really, really cool.
I like it.
Because sometimes I feel in science people do what they can do instead of doing what they really should do. And what they can do is very often determined by technology and availability of funds. So having the time sometimes to stop and really think back and reevaluate is very important.
Definitely. And I think this funding crunch has forced a lot of scientists to think really creatively in devising their next grant and different collaborations they can form and leverage to really make their application more competitive. So fantastic quote there. And now you mentioned Rutherford as one of these scientists that you admire. Can you think back on your career and tell us about some of your role models along the way?
Yeah, that's a great question, too. I grew up essentially as a physicist, not as a biologist. In fact, I became a biologist about 25 years ago. But my early doctoral studies, I was doing physics. And my absolute role model at the time was Richard Feynman. And it was Richard Feynman because here's a scientist who's been able to achieve the highest level of accomplishment while remaining a really emotionally complex and rich human being, and being able to bring forth a passion for knowledge and discovery that has been rarely seen in other scientists. So that's the kind of person that I strive to be, maintain the side of my humanity while also being able to understand how things work.
Fantastic example. And I think even a lot of non-physicists have turned to Richard Feynman for inspiration throughout their careers. So fantastic example. Well, let's rewind a little bit farther now. So can you tell us, Andrea, where your passion for science began? Can you share one of your earliest memories about being excited about science?
My family is actually almost completely in science. About 2/3 of them are in the medical sciences. My grandfather was chair of pathology in Naples, and many of my uncles are oncologists, etc. But my father was a chemist, and so became the kind of the black sheep of the family because veered off from studying medicine. And that helped me develop an amazing passion. Even when I was probably 10 years old, I had a chemistry lab that had very little to envy to an actual professional chemistry lab. I used to go to my father's lab and take every possible piece of equipment that I could get. And so I was doing pretty sophisticated chemistry, and I was studying at the time, I remember, quantum mechanics on books that really were not age appropriate. In fact, I really wanted to be a biochemist. And in Italy, at the time, biochemistry was not really well developed. And so I decided to go for physics to kind of more of a broader background, and then moved almost sideways until I could actually achieve what was my original aspiration.
So did you feel a lot of pressure when you were growing up sort of in a family surrounded by scientists to pursue this route or to excel in this route?
Oh, nothing special. I would come home with an A+, they would say, well, that's expected. If I get an A-, they would tell me, no, something is seriously wrong. We have to figure out what...
You're going to have to study a little harder.
Yeah, that's
Gotcha.
It wasn't exactly easy, but there was a lot of motivation.
So you have had a really interesting path and have done some different things along your journey. And I think the first one I'd like to chat about is this transition from your PhD in Florence, Italy, then to coming to do your postdoctoral studies in the United States at MIT. Was this a difficult transition for you?
No, it was a phenomenally useful and sought-after transition. It's almost an obligated path for any young physicist to spend some time abroad in labs that are sort of at the cutting edge. At the time, I was very interested and excited. My thesis was on chaos theory. This is a theory of not what you call stochastic chaos, where a lot of things are moving together, but rather a theory of deterministic chaos, which is one that arises when you have even very simple model. Like, for instance, you can attach one pendulum to another pendulum, and simply you cannot predict where it's going to be after about 10 minutes of oscillating. And also I thought that chaos theory was very appropriate for an Italian, so I know that was part of my studies. But then when I went to MIT, I started working on something called information mechanics, which has become then the dominant component of my research as a biologist. And so I would say it was probably one of the most exciting moments in my career, but I was also 1 month after I got married to my wife, and it was certainly challenging to move from Florence into a completely novel place. But we adapted.
Get all the big life changes out of the way all at once, right?
Right.
Well, fantastic to hear about your experience. Now, we mentioned in our introduction that you've co-founded and founded different companies. Can you tell us a little bit about that experience and what really drove you into that entrepreneurial path?
Well, it's actually interesting. Founding a company is not really about trying to make money or trying to become rich overnight or riding the web bubble. For me, it was more the ability to actually implement on a practical scale something that you do in an academic lab. And I really consider what I was doing at IBM to be completely academic. In fact, probably IBM has been one of the most academic places I've ever worked at, because there was absolutely no pressure for developing any kind of product or tangible thing. It was all about the science. But the level of science that you could do lacked, I think, some practicality. And I think that having companies in place really helped me to understand a little bit better what it takes to actually really deliver the results of your science in a context that can actually make it useful for humanity.
Certainly. And I think one of the complaints that you hear from a lot of scientists is they're not necessarily trained in a business sense. Was this something where you found you were developing a lot of the skills along the way, or you had mentors to help you through this process?
I think I walked right into that. In fact, having spent 13 years at IBM helped, because while I was doing fundamentally science, there was also a component to the business side of IBM that I was exposed to. And actually, I had started a company even before coming to the States. When I was in Italy, we had a company that was really an advanced computing company at the time. In the 80s, there were not that many computers that were more powerful than today, maybe a watch. But it was very exciting because we helped develop some very large systems. And so I kind of have a knack for it. And I tend to be pretty structured in the way I organize work and sort of delegate. So that has been a very, very useful experience, even in terms of then later on running some large centers of excellence here at Columbia.
Well, fantastic to hear more about your journey. And can you tell us a little bit more next about where you are today? Describe one of these projects that you're working on now that you are the most excited about, Andrea.
The project that I'm most excited about right now is about changing the way we think about precision medicine. And right now, the paradigm for precision medicine is based almost fundamentally on this idea that you can discover mutations in disease, and once you know the mutations, you will actually figure out what needs to be done on an individual mutation basis. Unfortunately, this has not always worked so well, and in fact, I would say that the cases where it has worked have been more the exception than the rule. For instance, let me give you an example of things that we have discovered in terms of the role of the mutation. We still have absolutely no idea how it even works or what to do about it. So in ALS, is Lou Gehrig's disease, we have known for a long time that familial mutations, such as mutation in a gene called SOD1, would be 100% probability of generating the disease. And yet, maybe 20, 30 years after discovering that, we still have no idea how to treat that disease. What we are trying to do today is deal with a very fundamental paradox that has been somewhat acknowledged by everybody, but also not really dealt with directly. If you look at the way cells express their genes, if you take a disease category, the cells without disease, for instance, basal breast cancer, will actually have very, very similar programs of regulation of their genes. They look almost identical. If you actually look at the genetics for that exact same set of patients, it's all over the place. So you would never be able to put them together.
Really?
So the question is, how is it possible? In fact, triple-negative breast cancer, which is almost overlapping with basal breast cancer, is actually defined. It's called triple-negative because it lacks the three most frequent mutations in tumors, which are the amplification of the estrogen receptor, the amplification of progesterone receptor, or of the RB2 protein, HER2, which is now a target of a drug called trastuzumab or Herceptin. But they lack those three mutations, and therefore there are literally no two patients with basal breast tumors that have the same genetics. And yet, if you take all the tumors that have ever been collected and you just look at their expression profiles, all the basal breast cancer would go right together. They would just be virtually indistinguishable in terms of their gene expression regulation. So what we did is basically try to figure out how this is possible. How do you solve this paradox? And we thought that one way to solve it, which we now demonstrated in a lot of mechanistic detail, is that in fact within disease there are some bottlenecks. You can think of them as funnels that essentially collect all the bad signals from any kind of mutations upstream and then implement downstream these programs that are necessary for implementing the disease in a homogeneous way. So even though you may have a completely different set of mutation, if those mutations end up being upstream of the same bottlenecks, then you implement the same type of disease. And this has been transformational for us because, first of all, it tells us that instead of drugging the actual mutations, you should drug these bottlenecks because they're much more independent of the way you implemented the tumor, and therefore they're less likely to relapse, more likely to have a single drug work for multiple people, etc. And more importantly, that we can actually discover these bottlenecks, proteins that we call master regulators of disease, by simply traversing back the regulatory network of the cell. And this has been incredibly helpful and incredibly successful, I would say, in the lab with 10 to 15 different tumor types. And now most recently, we've applied the same type of approach to study ALS, as I said, Parkinson's disease, Alzheimer's, even alcohol addiction. So this has been a really exciting journey, and I think something that is quite different from the way we think normally about dissecting disease mechanisms and then targeting them pharmacologically.
Well, this is fascinating. I myself am in research in Parkinson's disease, more on the neurorehab side than the genetics and mechanistic side necessarily. But I get a lot of questions from the public, from people I know, how close are we to finding a cure? Is this something that you get this question a lot, and do you have a good answer for it?
Yes, the answer is that we are every day close to find a cure for a particular type of cancer. For instance, very recently, we went from having melanoma be an almost incurable disease once it metastasizes to having long-term response in a substantial fraction of the patient by using immunotherapy. That has been a major breakthrough, and I think, for instance, immunotherapy, which has always been the prodigal child of cancer biology, it was on since the beginning, but it never quite really worked the way it was supposed to work. And it finally, through some of the work that Jim Allison did, and some of the work that Carl June did with CAR therapy, Rosenberg at the NIH, we're finally starting to see the light at the end of the tunnel, and it's starting to produce some pretty spectacular results in a number of tumors that we thought were untreatable. But unfortunately, diseases like cancer have the problem that they're extraordinarily heterogeneous, and therefore there's really no one key fits all locks. So when you say, when are we close to finding the cure for cancer, it's a bit of an oxymoron because potentially there are as many different cancer as there are individuals with cancer on the planet, probably as many different cancer as there are cells within individuals, because even different cells within tumors have different mutation and different type of properties of resistance to drugs, for example. Mm-hmm. So I think we are using a combination of three different branches of biology. The first one is traditional oncogene dependency approaches, which target genes that we know are very important for cancer, such as KRAS, EGFR, HER2, etc. The second one is immunotherapy, which has now really started to pay big dividends on the earlier work. And then the third one is, as I said, this kind of new branch of we call cancer systems biology, which looks at the cancer cell as a complete system of interacting parts. And it's trying to figure out what are the key vulnerabilities. And very frequently we find that it's actually not just one gene, so you cannot discover these by one gene at a time, but are two different proteins that work together, and they only work when they're together. For instance, very recently we showed that in aggressive prostate cancer, this was a collaboration with Corey Badashen and Michael Shen here, that there are two proteins, one called FOXM1 and one called CNPF, and only when these two proteins are co-expressed, and every time that those two proteins are co-expressed, you get the most aggressive prostate cancer malignancies. And in fact, you can use these as spectacular biomarkers in a cohort of about 900 patients that were processed at diagnosis for the presence or absence of these proteins. You could predict up to 20 years in advance that the people with the double negative, both of these proteins inactivated, would actually never die of disease. Only, I think, 3 out of the 900 died of, we don't even know whether it was prostate cancer or something else. Instead, the ones that had the double positive were about 90% of the people that actually died of prostate cancer.
Oh, wow.
And the single proteins had absolutely no predictive activity. So you would never be able to discover these one gene at a time. But this was only possible because we figured out how the machinery that makes the aggressive cancer tick was working and basically told us these are the two proteins. We went to the lab and we showed that those were the two proteins.
Well, Andrea, you are doing some phenomenal work there at Columbia. Great to hear a little bit more detail about your research. But I think so far we have made your experiences in science sound very smooth and easy almost. But I know scientists face so many challenges today, and these challenges come in a variety of different forms. So can you think back over the course of your career and share with us one of your own challenges or one of these failures that you experienced at some point?
So I'm a merry-go-lucky kind of guy, and so when I find a challenge, I don't even look at it as a challenge. I just start doing something else and move aside. So I never really felt that there was something that didn't pan off and that was a tragic or a problematic point. But probably the most challenging moment in my career has been when I moved from industry to academia, simply because the funding model was completely changed. And so I arrived at Columbia as a full professor without ever having written a grant. And I remember the first year and a half, I didn't quite know how to exactly do that, because when I was at IBM, you would simply do your science, and based on how good it was, they would decide whether to fund you next year or not.
Gotcha.
It was actually made a lot of sense. But then I figure out that you have to work more in an environment where you have to propose your science, and then somebody else who might not even know about it can tell you whether that has been too ambitious or not. So the first year and a half was a little bit of a struggle. There was anxiety about whether this was going to happen or not. And then literally 1 year and a half after I joined, essentially pretty much all the grants that I proposed went through and were funded. And that was the start of a really very exciting and very prolific part of my career. In fact, we just completed 10 years of this center that was funded right after I joined Columbia, that I was the PI for. And this is a center that had a 10-year lifespan predefined. So renewable after 5, and we did get a renewal, but it was quite spectacular because I think we had something on the order of about 400 publications between the group.
Oh, wow.
1/3 of them in very high-impact journals. And it was not just me. It was a group of maybe 20 investigators. Quite spectacular experience, and that taught me to work in groups in collaborative fashion, which I think is really now becoming a hallmark of the type of research that I do.
Definitely. I think the collaborative aspect of science is becoming more and more important over the course of the years, at least that I've seen. And I love hearing this story of taking a challenge and turning it into a success. So can you tell us what are some of these key things during that initial kind of rocky year and a half that really helped you turn things around? Was it just your persistence in continuing to submit things, or did you have certain people help you along the way?
I think it was a combination of knowing how to tell a good story. Sometimes it's funny, but in science, knowing how to write English is even more important than having good ideas. But I would say it's a combination of having good ideas that we believed in. There were ideas that at the time were really controversial. So we were proposing to be able to reconstruct the entire logic of the cell, starting primary data from patients. That was never done before. It wasn't clear that it was ever going to work. And our first paper in 2004 was the first network that was reconstructed from human cells and was experimentally validated. That was really a milestone. But I think the other ingredient, so the third ingredient, was the ability to find the right people to work with. One of the things that I really love to do is to learn. If I stop learning, I just kill myself.
It's over, yeah.
Every day, has to be a new learning experience. Otherwise, you fossilize and you become useless. And of course, there are increasing challenges as you go on in life. But at the time, studying as a physicist and somebody with strong mathematical and computational background, the thing that I really wanted to do is to actually learn and understand biology. And I think Ricardo Dalla Fabra here at Columbia, who then became the Cancer Center director, was absolutely critical and an unbelievable help for me in sort of making that transition happen.
Gotcha. Well, I think you brought up so many great points, Andrea, and I love this description that you have of your love of learning. I think this passion to continue to learn, to continue to grow, and to continue to know more about the world is something that drives a lot of scientists. So very cool to hear about it, and great to hear about some of these successes along the way. And I love sharing success stories. So do you have a favorite one, perhaps a recent success that you'd like to share with us today?
Scientifically, the thing that has made me most proud has been a recent publication that we had in Cell, where we actually finally able to demonstrate this idea that these bottlenecks exist, and not only exist in terms of having a few proteins regulate the actual behavior of the cell in disease, but that you can walk upstream of these bottlenecks and discover systematically all the mutations that are relevant for cancer. And so what we did in that case was, in human glioblastoma, there is a subtype that we had previously published in Nature in terms of identifying these two genes. Again, this is another example of two genes where one doesn't do anything, the other one doesn't do anything. You put them together, you get the most aggressive type of glioblastoma. And so we knew that those were, in fact, the experimentally validated bottlenecks. And so what we showed in this paper is that despite multiple efforts by a lot of really amazing groups in trying to figure out what the genetics of this disease was, the genetics of the mesenchymal subtype was essentially still completely unknown. About 30% of the patients had a potential putative explanation, but not mechanistic one, with a gene called NF1. What we did is we essentially walked upstream of these three proteins, CV-beta, CV-delta, and STAT3, that we had discovered and previously published using computational algorithms, and were able to identify mutations in genes. One was actually CV-delta itself, so that was not a surprise, but it could not have been found by conventional genetics because the actual probes on the genomes were not dense enough around that area to be able to show that event. So you have to go and really look specifically for that gene to be able to find it. The second one was a protein that was deleted in about 60% of the mesenchymal subtype patients. And so it was remarkable that people could not have discovered that because it's so frequent. It turns out that the reason it had not been discovered is because it's sitting on the genome right next to one of the most famous tumor suppressors called P16. And so when people look at that region, they immediately thought it was P16 and not these proteins that there's only 4 papers for in the entire PubMed. And so, but the computational algorithm told us that that was actually the protein. We were able to show then that this actually was responsible for capturing the master regulators and tagging them through a mechanism called ubiquitination for degradation. And so when this protein was deleted, these master regulators were going completely out of bounds at the protein level. They were not changing in terms of expression, but were accumulating at the protein level, and then were driving this very, very aggressive phenotype. We then showed that the same thing could be done for breast cancer and even for Alzheimer's. And so I think that has been something that I've always longed to do for basically 10 years, to show that not only we could find the bottlenecks, but actually that the bottlenecks were really capturing the mutational load of the disease. And I think the fact that this can be applied both to cancer and to non-cancer phenotypes has been quite exciting, and it's now become the complete basis for what we do in precision medicine at Columbia.
Well, congratulations on this exciting success and really contributing to a big advance in the field, it sounds like. So very, very exciting. And I love to chat about research. And we talked a little bit in the beginning of our interview about some of the stuff that occupy your time when you're not doing science and research. And you mentioned that you like to read. And one of the things that I love to do on our program is get book recommendations from our guests. So, Andrea, can you share with us a recommendation that you have for what we should be reading next?
Sure. So I'll give 2 recommendations. One from my childhood. When I was I think about 14 years old, I read Hermann Hesse, Narcissus and Goldmund. Most people know other books by Hermann Hesse, but this one is quite spectacular because it's a story of a young boy that is completely intuitive in nature, has absolutely no rigor, but incredible passion for life, and who becomes friends with an abbot who is the exact opposite. He is completely rigorous. He has absolutely no flexibility, but also extraordinarily motivated in doing what the thing. And I think that for me, it's been almost a parable of what my internal mind state is, because I have a very intuitive, tactile side where I try to really play with things, but then at some point I need to think of them deeply in a way that allows me to be rigorous about them. And I constantly struggle with whether to give in on the more intuitive side of the character, more effervescent or ebullient side of the character, versus the one that is more rigorous and maybe a little more sedated. And I think that struggle has been very helpful for me in life. The second one is a book by Neil Stephenson called The Baroque Cycle. It's a trilogy. It's an extraordinary set of books, mostly because I think Neil is one of the most probably underappreciated American authors. He has the ability to write that reminds me of James Joyce. He can write sentences that are literally half a page to a page long. And
that's skill, yeah.
No, not punctuation. And the story is a story of one person that is called from the new continent, so this in the 1600s, from the United States back to England to mediate a litigation between Newton and Leibniz, on who invented calculus, of course. And it's played on the historical background that is incredible, from the plague in London to the wars in Europe, to pirates on the Barbary Coast, etc. And I think it is one of the wittiest, funniest, and most intriguing, and in terms of the background, historically correct books that I've seen.
Well, fantastic recommendations there, Andrea. You've definitely sold them well. I will put them out on our website for our listeners to find if they're looking for their next great read. And during our introduction, we mentioned a couple of the different places where you've been to do your science. And I think scientists get to travel quite a bit as part of the job, not only for the different positions that they take, but to go to conferences and other opportunities. So can you think back on all of the places that you've gone for science and tell us about your favorite?
Sure. It's hard, but at the same time, actually, it's a quite simple choice. Last year, I was invited by the Ipsen Foundation, a conference in Paris called Biology Seen Through the Prism of Cancer. And I grew up in Paris when I was a kid, so it was extraordinary for me to go back for a bit of an extended period of time there. But also, this was just an extraordinary event. We were first in Paris at a private visit of the Louvre that was shut down at the time, so it was almost all to the group of people there. And then we were for like 4 days captured in a chateau in Chantilly. So that sounds very glamorous and exciting.
It does.
But I think the most exciting thing was that the group of people there was anybody who had done major discoveries in cancer, from Mike Bishop to Luke Hadley to Inder Verma, David Baltimore, Bob Weinberger, Phil Sharp, and Al Varmus, just to name a few. And each of us had about 30 minutes for presenting and then 30 minutes for just broad discussion with the group. And it's been a format that has been so exciting and intriguing. I have extraordinarily fond memories, in particular, actually, of meeting there Inder Verma, who is an amazing human being and cancer scientist.
Well, this sounds like an incredible experience and a fantastic opportunity. I know I've been to Paris once before, and the visit to the Louvre just really stuck in my mind as phenomenal. I mean, you're surrounded by centuries of beautiful art. I can't imagine sort of having that place to yourself or with a small group of people to just explore everything. That sounds incredible.
I grew up in Florence, and so I was surrounded by art the entire time I grew up, so I became a little bit numb to it, and actually then in Paris when I was 14. But going back to it allows you to rediscover it in a way that you had never looked at it before. There's nothing like deprivation to make you appreciate things.
Certainly. Well, thank you so much for sharing this story with us today. And I think you really touched on in discussing some of these opportunities that you had as part of this experience to talk to these people, to be in these, relatively small group sessions with experts in your field. And I think the people that you get to work with and interact with as a scientist really make it a fantastic job and a fantastic experience overall. And I've worked in a couple of different labs over the course of my career, and each lab has its own personality. And it's fun to talk about the different traditions that different labs have, or even just some of the fantastic memories that you guys have shared together, and kind of highlight the human side of science. So, Andrea, can you tell us about one of these traditions or one of these fun memories that you have about a lab that you've been in?
I struggle with that a little bit, thinking about that question. Actually, have a group of people that is so heterogeneous. We don't really have this kind of clear tradition. But I'm going to share a couple of funny moments in science that I think you'll enjoy. The first one is that my name is Andrea, and it's the only country in the world where it's a male name is in Italy. Actually comes from Greek, and it means man, so it shouldn't be used for a woman, but that's a different story. So,
Gotcha.
I'm probably the only male scientist that has ever been nominated for Women in Science, so that was a very exciting moment of my career.
Congratulations on that nomination.
And I've always had trouble when I go to conferences because I always get paired up in the women's dorms, or, once I was Mr. and Mrs. Tolovitsky when I went there with one of my colleagues. Anyway, so that was one. The second one is that it's my experience as a comedian. So I gave a talk at the American Society for Human Genetics in Philadelphia. This is the annual meeting in 2008. And there was a time where the computational biologists and the experimental biologists were still a bit of a dog, they're starting to learn about each other. And so I told this joke about the computational biologists and the experimental biologists on death row. They're asked for their last wishes. And the computational biologist says, well, I would love to have opportunity to present my work to an audience of biology because they never want to listen to me. They all want me to look at their data, but they never want to collaborate. And so the executioner says, well, of course, tomorrow is the plenary session of the AHSG, and you'll give a talk at 12, and we'll kill you at 1. So the guy says, thank you, this is fantastic. It's a coronation of my lifelong dream. And then they ask the experimental biologist what the last wishes are, and he says, could I be killed before 12? And it was very funny because there's about 7,000, 8,000 people in the audience, and you can't see anyone. You have lights in your face, and
right.
Just seeing. And the audience just literally erupted. So I felt a little bit like Seinfeld, it was being at a comedy show. So that was a lot of fun.
Oh, that sounds like a fantastic experience as well. I'll get a little laugh from the audience. And I myself, as an audience member for many of these talks, do appreciate bringing in humor. So thank you for that. Well, I love highlighting these really fun and funny moments in science because I think there are a lot of stereotypes out there about what the life of a scientist is like and what scientists are like as people, just sort of being very serious and kind of working in your own personal endeavor tucked away in a laboratory. And I think that's very different from the experience of a lot of science today. So we talked about some of your current research, Andrea, and some of the questions that you've answered over the years. But one question that I love to ask everyone on our show is about the questions they haven't yet been able to answer. So Andrea, can you tell us if things like funding and staff and technology and feasibility, a lot of these major barriers in science today, if they were not an issue, what is the question that you would like to answer?
So the question that I'm totally fascinated by right now is both an important question, in fact, I would say a crucial question in cancer, but also more generally in human biology, is the notion that now the cells live in sort of an equilibrium, it's called plastic equilibrium. They go and develop into lineages that then building the parts of our body. And we are starting to unravel the regulatory networks that allow us to think about how to reprogram cells from one lineage to the other. So the question that I really would love to ask, and that I'm starting to ask right now, but it will require some significant amount of time and of funding, is basically how can we modulate cellular reprogramming using only small molecules? So do it in a way that would be not invasive, but would allow us to regenerate any kind of tissue we want, will allow us to study any particular tissue subtypes we want by just regenerating from iPS cells. And some of the early work that we've done, as I said, they're just being published in stem cells, suggest that this approach that we've taken for cancer can actually be successful in the area. In fact, I would say probably the very first work that we did using regulatory models that was highly impactful, in fact, I think it was one of the top 15 most impactful paper in 2010, has been showing that the two master regulator I was telling you about for glioma and STAT3 can reprogram neural stem cell into mesenchymal cell. And one other gene that we discovered, called NF238, was then later on rediscovered by the group at Stanford that actually reprogrammed fibroblasts into neurons. So the cells are in states, but the states are very plastic and they can be changed. And now we're starting to learn how to actually do that. And I think that's going to have a humongous impact in terms of regenerative medicine, but also one of the best therapeutic approaches in cancer has been differentiation. If you can differentiate a tumor cell into a terminally differentiated cell, that tumor will essentially not be able to renew itself and will eventually die after about 10 to 15 cycles. For instance, the leukemia called APL, the treatment with retinoic acid, which essentially forces the cell to differentiate, has been completely curative. And I think this is a strategy that may actually play out in a variety of other tumors.
Well, Andrea, I think you have gotten my mind ticking and our audience members as well. I think these methods, these techniques, these emerging technologies in regenerative medicine, particularly at the cellular level, have so many exciting applications in science and medicine. So very cool to hear your thoughts on those. And we've talked a little bit about your journey. And I think one of the things that we haven't yet highlighted is some of the great advice that scientists get along the way. And this might help them really propel them to where they are today. So can you share with us what you think is the best piece of advice you've ever received?
So I've been given a lot of great advice, which I've never listened to. But one that I actually did listen to was, I think, sort of a second-hand advice from Eric Kandel to Richard Axel. I think the story that Richard told me was that when he was a young researcher and Richard was already more senior and established, he was doing a lot of different things in biology and really spending time solving a variety of different problems that were all very exciting. And Eric told him, why don't you stop fussing around and concentrate on one thing and just do that one thing and focus on it and do it well. And I think that advice is very important because you may have seen from my career that I have sort of moved from physics to then computer science, from computer science to computational biology, from biology to chemical biology, and now I'm working a lot on sort of clinical and translational sciences. So the field is changing quite significantly, but I think what I've done that has really changed from my early years where I was actually just going wherever the wind was blowing and I thought my methodology could work, to actually trying to really formulate a very coherent framework that is really centered around cellular regulation and tries to understand biology using that mechanism, and that mechanism is built using something that I've learned from physical information theory. And so this has been really something that has allowed me to, first of all, understand what are the genes that drive disease, what are the upstream genetics that drive the aberrant activity of those genes, and now most recently to actually figure out what are the drugs that can target these mechanisms in a completely different way from what we normally do, which is more empirical. And so we try things also based on some hypotheses that are reasonable, and we have sort of gone to something where we actually, what we do and where our intellectual activity is creating models. And here's another very good quote that says, no model is correct, but some models are useful. And so the idea is that you can use models today to generate a variety of hypotheses and then go to the lab, and when you validate them, about 80% of them are actually true, which is, to me, extraordinary and devastating, because even 5 years ago, I would have never suspected that for biology we could do something like that.
Absolutely. Well, I think this is great advice, especially early on in your career. I think having the ability to focus on something is so important and get something done. It's so easy to get distracted by different interesting questions that will sort of pull you in different directions from your main focus in a laboratory. So fantastic advice. Now, is there any other last piece of advice or last piece of inspiration, Andrea, that you want to leave our listeners with today?
Well, I would conclude by saying that people need to find a way to renew themselves in life on a constant basis. Sometimes we do that by having additional children. So after the first batch was almost out of the door, we had another 2, and that certainly keeps us motivated and going. But scientifically, I think you need to constantly question whether the actual problem you're working on is really relevant or not. And a lot of people, what I found, and I think is a big inefficiency of the system, is that a lot of people work on problems simply because they ended up working in those problems, because of the lab they started in, because of a twist and turn in their fate. And so they keep working on problems that in some cases are potential dead ends or potentially not really going to have major impact, simply because they know how to do it, they've done it before, they feel comfortable in that area. And I think at some point you have to make big decisions and really take a step back and look at what you're doing right now and take a risk and basically say, look, this is great, but I need to really formulate the kind of problems I'm interested in. And the people that have done that have typically done very well. There's a lot of cross-disciplinarity right now in science, and the people that have been able to be more effective at exploiting that have done really well. Jim Collins or George Church, Stan Leibler, et cetera, really have set us as role models for the way this should be done. And the reason I'm saying that this is a problem is because I think there's an incredible amount of talent, and it's not always used in the most effective way. And I think that if people question themselves a little bit more, they'd be able to go back to the drawing board, maybe take some risk, in some case fail, but in some case maybe start doing things that are much more exciting.
Definitely. Well said. Great advice there. I love this idea of constantly questioning and always questioning your own work and making sure that you are doing the work that you can do to make the biggest impact on the world. So fantastic note to leave our listeners with. Can you tell our listeners how they can reach out to learn more about you and your research?
I'm really easy to reach out because my name is a little bit unusual. And so if you search for Andrea Califano on the web, the very first hit is going to be my website where you have all my contact information, and I'll be happy to answer emails.
Perfect. Well, Andrea, thank you again for joining us today and sharing some fantastic experiences with myself and our listeners.
My pleasure.
Absolutely great to chat with you today. And thank you, listeners, for joining us as well. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.